words.
Yeah. Okay.
So now I'm
Yeah.
just sharing this window, but you can see it.
Yeah, we're good. Okay, so let me start it. So welcome to the Ozeki Journal Club. I'm Carlos.
Hey,
Barry.
Carlos.
Hey, Barry.
Yeah.
That's Barry. And today we have a guest, which is very nice. We have Florian Wünnemann. Did I pronounce that correctly?
That was perfect, Carlos. Hey guys,
Thank
how's
you.
it going? Glad to be here.
Florian, thank you
Yes.
for joining. So Florian, I guess, is a personal friend, mutual friend for me and Perry. And you are involved in research, I guess. You're a postdoc, right?
Yeah, so currently I'm a postdoc at the lab of Guillaume Lettre at the Montreal Heart Institute.
Mm-hmm.
And previously, I've done my PhD with Gregor Andelfinger at Saint-Justine in collaboration with the University of Münster in Germany. And yeah, the paper we'll be discussing today is part of my big part of my PhD work, actually.
Oh, it's PhD work?
Yeah, so this is like the biggest paper, I think the biggest project I've been working on during my PhD.
So
Nice.
first author, congratulations on that, on Nature Genetics.
Yeah, that's
Very impressive.
insane. I mean, I wanted to ask, like, how does it work to be the first author on a paper with, like, I don't know how many that is, like 20 authors? Like, how do you deal with that? I think because Perry and I both come from fields that usually have like maybe 5 authors when it's a lot. Yeah, I was curious, like, what's that experience like?
Yeah, so like, first of all, let me say that because there's a lot of people on this paper, it's like really a collaborative effort, right?
Yeah.
Papers like these is a lot of work, and I certainly couldn't have done that by myself. And so like, credit to everyone who's on that paper, like all the co-authors, my boss, like everyone has worked really hard on that project and, trying to get the best data out of it. And so like, that's the first thing I want to say. And then how is it to work? So it is challenging because obviously there's like a lot of different, like it's a very, like a lot of different areas that we worked on, a lot of different types of experiments we did. It's a combination of wet lab work, bioinformatics, clinical work. So there's medical doctors involved, there is bioinformaticians involved like me, and then there's people involved that do wet lab work as well. So, it's hard to coordinate sometimes, but you talk to people, sometimes, that you don't know how to do a certain experiment in your lab, or you don't have the expertise, but you know someone who does. And then usually, you contact them and see whether they can help you with trying to do the experiment you think is necessary to answer a specific question.
How do you decide who's first author though?
Well, usually I think it goes mostly by who has done the most work. And
okay.
But isn't it you that like designed the experiment, but like the whole project, right? Or someone, and then you say, okay, well, I need help on these experiments, and then those people get added to the paper?
Yeah, so I should also say we're co-first authors on this paper. So
okay.
my co-first author is Asaf, and that means we've kind of like, I guess, contributed equally to this in terms of workload, in terms of like, and then, so your question was, Carlos, sorry, like already.
Well, I think maybe I followed up on Perry's question, like, how do you decide, how do you conceive of that kind of research that's going to involve so many people, right? Like, I would imagine you and your co-first authors and maybe the last author, like the supervisor, have like a target question they want to answer, and you kind of supervise, or like you coordinate, I guess, the whole effort, I would imagine.
Well, so in this case, it was a project where we had this lead on this family that was seen, that we had a genetic like lead there, and we wanted to investigate whether that was the causal gene that was like responsible for their disease. That's kind of how it started, and we had some molecular biological data, and then from there on, it was just like I joined the lab and kind of became responsible for this project, and then. Yeah, from there we kind of just conceived several experiments to see, okay, what can we do? For example, you'll see in the figures in the experiment we've done, we've created like a transgenic mouse model. That's one of the experimental models in cardiovascular research that we can use to see whether a gene is involved in heart disease or in congenital heart disease. Yeah, and then from there it kind of we've designed multiple experiments to answer the questions that came up once we. So it's not, I know what you mean. I've checked some of your other journal clubs and I know the kind of research you guys do. So it's not this like idea where you conceive a very big question in the beginning and then you know what you have to do and you do it. And at the end you have a very definitive answer. This one is more of one of those projects where you have an interesting lead and then you see whether whether your hypothesis in the beginning is actually true, and to what degree you can prove that, I think.
Mm-hmm. Okay. And your background, you said you did mostly bioinformatics work in this?
Yeah, so I have my degrees in biology, and my training has also been a mix of bioinformatics and wet lab biology.
Mm-hmm.
So during my PhD, I've done like 50-50. So I've been 50%, like more or less 50% in the wet lab, and I've been on the bench and I've done experiments, but 50% I've done bioinformatic analysis. And that's certainly more where I think my passion lies, is bioinformatics, and pondering with the idea of doing computer science before biology. But decided to go into biology. And so now in my postdoc, I'm really 100% bioinformatics, and I don't do experiments at the bench anymore.
Mm-hmm. Nice. And you're looking for faculty positions, right? I think Perry told me.
Well, I haven't started looking yet.
Okay.
I'm still in my postdoc, and I'm quite happy. By the end of by middle of 2021, like end of 2021, I'm looking for new positions.
Mm-hmm.
And I'm considering faculty positions, applying for those, considering doing another postdoc. We'll see.
Mm-hmm.
I'm not quite 100% sure myself yet, but
did you enjoy the postdoc experience? Because I'm looking for postdocs now, I would say. I like enjoy research, but I don't know. I guess I've never spoken to many postdocs. I didn't have many postdocs in the labs I've been in. How did you find it worthwhile?
Yeah, definitely. I really enjoy it. It's very similar to the PhD experience, I would say, without the stress of having this big exam at the end, and without the stress of having, like, that's at least, and I think many PhDs have this kind of fear, anxiety. Pre-doc exam is pretty stressful, and then you're working towards this big goal at the end, right? And it's this like 4 to 6 year journey or 7 year journey.
Yeah.
Depends on what field you're in.
I mean, I'm like a bit. I think obviously the next thing you take is always going to feel like it's the most stressful thing. But I would imagine like going into a postdoc, it's almost like they're hiring an employee at that point, right? So it's almost like you are being paid to deliver a service, and you're assumed to be like an expert in a specific topic, and that's why they take you. Whereas in a PhD, it really feels like, you're responsible for your crap. If like it takes you a long time, it's on you. No, you're kind of like a student still, so you're in a sense still learning, and maybe you're paid by some like scholarship. So I feel like a lot of the responsibility falls, all the consequences end up on you. So I feel like I would imagine being hired in a postdoc lab as like, okay, well, Carlos, what can you do for us now? And I feel like I'm not really, I don't know. I guess we'll have to see how it goes, but that's the impression I get.
No, I understand where you're coming from, and I feel a kind of sense of delivering too, of course.
Mm-hmm.
But it's not like, it's still, you have projects and you still discuss them with your team or with your supervisor, and you still kind of come up with ideas, and you still learn new things. It's not just like you're like an employee and you're delivering analyses and certain goals. Like, sure, you have goals and you have timelines, and you try to keep to them. But as research goes, it usually takes longer than what you think it takes, and just because you're an expert in a certain area doesn't mean it all becomes suddenly much easier or faster. Like, certainly not. Like, again, even as a PhD, I feel people are already, because they read so much about their area of research, and they spend so much time on it that they're kind of experts in it. And having or not having the degree, I don't know if that, you don't change the day you get the degree. You don't become 20% more intelligent or something.
Yeah,
like you're the same person. So.
Yeah, for sure. Okay, well that's a nice insight. I'll make sure to keep that in mind. Okay, so let's start on this paper. It's our first I guess it's our first life.
It's first bio paper.
First bio paper. First anything to do with life, I think. Living things. Actually,
sure.
Yesterday we did one kind of on social science.
Oh, of course. That's even more life.
Yeah, I guess so. I don't know if you saw our last one that just came out yesterday, but we read a paper on how like racial and gender diversity in PhD groups is correlated with like creativity of the PhD or innovation of that group. So yeah, I don't know. It was an interesting paper from PNAS.
Is that paper where they kind of what attracted a lot of attention on social media, where they came to the conclusion that, like, which was horrible, and I think probably shouldn't have been communicated that way, where it was like female PIs have a worse impact or something? Was that
No,
a
that
different
was
paper?
another one. That was another one that
'Cause-
maybe we should look at. That was, I think, more recent.
We're
And
becoming
I
a
think
life podcast.
they tried to, I think they tried to like retract that paper or something. But I didn't really follow it closely.
Yeah, it was one of those papers where, like, on social media, so it's a different paper that I'm talking about. But yeah, there was a lot of attention to it recently, and it had some conclusions that were pretty damaging to, sounded very bad in terms of what they were trying to say, I think.
Yeah, this one was definitely more in line with like the political correct agenda, like sort of dogma. But I think it still had some interesting analysis. No, but okay, now we're going straight to pure science, I think, which was really nice. I think it was, so I did my my bachelor's in computer science and biology together, and then I did a master's in biology. So I was kind of used to reading bio papers, but it had been a while. But it's nice to see like a paper talking about like the real world. I think I've been reading a lot of like theory and sort of more mathematical things that you can kind of just like invent. But here we're talking really about what's going on in the real world, which was nice. So I think maybe I'll let you sort of introduce the motivations and we'll just go through the paper like that.
Yeah, so just to tell the title of the paper once, which is Loss of ADAMTS19 Causes Progressive Non-Syndromic Heart Valve Disease. And so in the paper, if I start out, maybe go through the figures and talk a bit about what we've done. So in this paper, we basically started off with two families, two unrelated families, in which we identified so-called loss of function, homozygous loss of function mutations in a gene called ADAMTS19. And if we look at family 1, and I agree with you, Carlos, I think it's nice that some papers have real relevance in real life, and they show some direct implications. And in this case, we're starting from human data, so we're really starting from observations that we have in humans, and then we will translate them to in vivo and in vitro models. So like I said, in this case, we're starting off with one family. If we look at family 1 here, you can see this family tree, and I don't know how familiar you are with like genealogical trees.
I was able to get most of this, but I think Perry might have some questions since he's a little less familiar with like the bio.
Even this,
stuff.
Even this loss of, what you call it? Loss of, I forgot the term.
Yeah, loss of function. So I'll explain what that means.
Yeah.
So let me try to walk you through this, and then you let me know if you have any questions afterwards, okay? So the plus minus here. So first of all, we're focusing on this gene called ADAMTS19. Genes consist of exons and introns, where exons carry the protein coding, they basically are written in this 3 nucleotide code, right, that will translate into protein coding sequence. So in this case, we've just made in this figure here, we're just representing like each number is an exon, because this data that we have for this family is so-called whole exome sequencing data. So what that just means is that you've probably heard of genome sequencing, and that is becoming cheaper and cheaper. In this case, what people used to do a lot and still do is not sequence the whole genome, but they only sequence the exome, which makes up only about 1% of the genome. The reason for this is that it's much cheaper to do that, especially a couple of years ago, it was much, much, much cheaper than genome sequencing. But for human disease genetics, many strong disease-causing variants are actually found in the protein coding sequence of the genome, so the exome. So this is exactly what had been done for this family. And what we're representing with the numbers is each exon. And what you can see here, so on the y-axis, we are showing the kind of read depth. So we've sequenced this, and then how many reads do we have on that exon? And you can see that it's kind of similar, it's around like 1 for each of those for those exons here. But the ones that are marked in red, these 2 individuals from this cohort had actually no copies, no reads on these exons.
When you say reads, you just mean like how many times they're in the genetic code?
No. So each of us have 2 copies of these exons, because we have 1 copy from our mother and 1 from our father. When I say reads, I mean we use a technology to sequence this. Mm-hmm. And these reads, they're basically just how often we read this from our sequencing data. So like, it's a standard thing in biology, in genetics.
Like how much it's expressed. That.
Yes, but we're not looking at RNA, we're looking at DNA. So just like, can we, it's like a qPCR almost, if you want to think about it that way. It's like how often, like what's the signal of this exon in the genome? And it should be the same as for every other exon if you don't have a copy number variation.
So these people have like a full missing gene from their DNA.
Exactly. Yeah, exactly. For example,
like a missing exon. Like they have the full gene, but one chunk of that gene is missing.
Yeah, the easiest example I can give is, so there are obviously, each of us have small copy number variations. That's what it's called in our genome. Think of trisomy 21. That is a copy number variation of the entire chromosome 21.
Mhm.
So in trisomy 21, you have like 3 copies of chromosome 21.
Mhm.
The same thing can happen for any stretch in the genome, that the small piece of genomic DNA is found in the genome 3 times, 4 times, 5 times because of copying errors during cell division or during embryogenesis. What can also happen is that you lose piece of your DNA because of these types of errors that happen during copying of your genome. That is called a copy number variation. And in this case, we found a copy number variation that basically covers this first 8 exons of this gene ADAMTS19 and the microRNA, which is this here, a microRNA that is just before ADAMTS19.
Mhm.
And in these people, what we actually found is that they have no copies of this. So these individuals. Yeah, Perry, go ahead.
So this is not just like a random cell. This is like all their cells in their body are missing this gene. Or these exons, I should say.
Presumably, yes. Yes, all of their cells in their exome, in their body are missing this. We don't know if all of the cells are missing this. This could have happened anywhere because we have tested this obviously from blood. So we know that in the blood.
I see. Okay.
But when you do this, yes, because of the fact that we know that the parents, and I'll go to that in the family tree here, like you said, we can be sure that it is all of the cells in their body because of the fact that it's inherited from the parents.
Okay.
But there could be a scenario where it wouldn't need to be all of the cells. In this case, it's all of the cells.
How did you arrive at this gene specifically as being linked to this disease?
So what we do is, like I said, for this family we did whole exome sequencing. So we've sequenced the exomes of all of these family members. Well, actually, there's 4 family members, these 2 and 2 others. And then you perform like a bioinformatic analysis where you look for single nucleotide variants, which are like mutations in the genome. And you look for these copy number variations.
Okay, so you went through all of their genes, all of their exons,
yes.
and then found the ones where there's abnormalities. Okay, cool. Yeah,
exactly. And there's more than just ADAMTS19. There's other genes, but if you look at a certain type of variation that you have, so it has to be something that would be very deleterious, meaning it would probably cause something that messes up your development. The frequency in the population, because this is it's not a common thing, so you would look for something that's rare. So doing all these prioritization steps, you can and doing the inheritance in the family. So if you look at the family tree, we can come up with an hypothesis of what type of mutation we're looking for. So these double bonds here, that just means that the parents are closely related. So these are second cousins. So they're consanguineous is what we call it. And so then they have these children here. This means stillborn, so died before birth. And they have these kids here, and if you look, when they're black, it usually means they have heart valve disease. So heart valve disease is a disease where we have 4 valves in our hearts, and if these valves dysfunction, so it means either they let too much blood pass through in the wrong sequence, or they're thickened, or the blood passes through too quickly, then we speak of heart valve disease. And these children had very early in life, which is not normal. So it's a disease that usually is associated with aging and degeneration. But in this case, this family, the children had this very early, which is very rare. And the fact that 2 children had it kind of, leads to a very high suspicion that it might be a recessive inheritance. Recessive inheritance means that you would need 2 copies of the defective gene or of the defective genetic element to be affected with disease. So as I said, we all inherit 1 copy of each gene, or half of our genome basically, from our parents. 1 copy of our genome from our mom, 1 from our dad. So in this case, the hypothesis was that the dad would carry 1 copy of this genetic element that's causing the disease, and so would the mom. And when we did that, we found, as I said, we found this genomic deletion of these first 8 exons of this gene. And when we look in the parents, both of them carry 1 copy of this deletion. So this leads us to the fact that we find this. And this is when we prioritize, again, the whole exome, this is kind of the only candidate gene we end up with.
And so the fact that the parents are related obviously increases the probability that the kids will end up being homozygous.
Yes.
For this, right? but I guess they're even second cousins, right? Is there like some sort of like relationship also? I thought second cousins were safe, basically.
Well, it's never safe, right? Because it just
one
depends.
eighth. It's just the probability of having the same genes, no?
The same mutations.
The same mutations, sure.
Yeah.
Sorry.
Because we all have the same genes. So yes. So what obviously, like, it's never safe, right? The reason why, like, obviously we say, oh, you know, consanguineous relationships are dangerous genetically is just much higher probability, right? Like you could, I mean, you can have theoretically could have kids with your first cousin and it could be fine.
Yeah, for sure.
In terms of genetics, it's just the probability is higher that there will be genetic defects.
Weren't all these kids being born like to like kings that were married to their sister, like in ancient Egypt or something? Or
yeah,
I think that was fine back in the day.
Royal families in the past, they used to.
But even maybe more recently, even
But so I think like second cousin is a pretty distant relation, no?
That's like 1/8, right? You have 1/8 possibility of having like I guess similar mutations that could lead to something bad.
Yeah. Second cousin is basically your grandparents.
Our brother and sister.
Oh, okay, I see.
No, so your grandparents are brother and sister, or brother and siblings.
Really
yes,
bad
exactly.
With this.
So it's 2 to the 3, 1/2 to the 3, because it's 3 generations removed.
Okay, cool.
Perry got it right, I think, with the math.
There you go. Now you also have the second family, right? Family 2.
Exactly. So we have a second family, and this obviously increases our like kind of how sure we are that this could be the gene. Because what I didn't say is, so like these so-called loss of function mutations. So there's a group of mutations or kind of like genetic aberrations that we call like loss of function. In this case, it's a genomic deletion, but you could also have something we call a stop gain mutation. So as I said earlier, like reads have an open reading frame, right? So they translate, the genetic sequence into amino acids by using 3 nucleotides. And there are obviously also trinucleotides that code for stop codons. So in this case, in this other family, we didn't find a genomic deletion, but we found a mutation in the gene that codes for a stop codon inside of the reading frame. So it basically causes the protein, well, it doesn't cause the protein to be shorter, but it causes the transcript of the gene to be much shorter. And at the end, what happens is that the transcript will just not be translated to protein. So the end outcome is that they can't make protein from this faulty gene, let's say.
Stop codon tells you to stop adding amino acids to the protein.
Exactly. So it just tells the transcriptional machinery. Well, it doesn't say stop adding amino acids. Actually, it says just stop transcribing here.
I think like what happens is like there's a protein that scans along the gene, and if it sees a sequence of 3 nucleotides that correspond to the stop, it'll just stop reading at that point, and whatever came afterwards won't get turned into protein.
Actually, I think it will continue reading, but then the machinery that checks this will see the stop and will say, oh, this is not correct, degrade this. Like, don't make, it's called nonsense mediated decay.
Yeah.
And it's, so I'm not 100% sure where the transcriptional machinery stops writing. I'm not sure it stops immediately at the stop. I think it continues a bit, but at some point there will be a machinery that's like, oh, I'm going to read these transcripts from the RNA and say, oh, if there's a stop codon in the middle or like somewhere where it's not supposed to be, it knows, okay, I should not put this out and make protein out of it.
Yeah.
It will just, because there are some other signals that are then missing in the genome, like a polyadenylation signal, which is required to export the RNA out of the nucleus. Lots of molecular biology, going way too much into detail.
It's so insane. No, but this is, I remember like, there's also mutation correction mechanisms, right? Like if there's certain proteins that look for parts of the genome that have the wrong letter, like the wrong nucleotide in them, and they go in and change it, I think. If I'm not mistaken.
So this is something that happens obviously in dividing cells a lot, because if you think about it, like cancer is just a cell that's gone wrong where there's a mutation that hasn't been corrected. And therefore the cell goes haywire and usually develops into something it's not supposed to. But in most of our cells in our body every day, this machinery will correct mutations and make sure that a cell doesn't become a malignant cell. Yeah,
that was, I think, the thing that got me really interested in bio, actually. Like seeing that in class, I was like, oh my God. How does it
I don't know
do
what's
that?
I don't know what's crazier, that this happens or that we figured out that this happens, you know?
That it happens, obviously.
I don't know.
It's a bunch of like balls of charges that like figure out when there's a mistake and they go and change
but
it.
we
You know?
figured out that that's like the mechanisms, you know, like
Yeah, for sure.
that's also crazy, right?
Yeah.
I don't know.
Yeah, I also find it crazy, and certainly this type of observation is also what got me interested in genetics and in biology, which is like, yeah, there's all these crazy things going on. You know, there's all these genes, and then a tiny mistake in one gene can cause you to have a super a very severe disease, for example.
Yeah,
I think that's crazy.
For sure. Okay, so
So yeah, getting back to the paper. So at the end of the day, the observation is that we have two families that have homozygous. So, you know, they're missing two functional copies of this gene ADAMTS19. In the end, what you can think of, these four individuals here that all have heart valve disease, very similar heart valve disease, they cannot make ADAMTS19 protein. There's no way that they would have functional ADAMTS19 protein ever since they were born. And then the other important observation for this is that this is very rare. This is a very rare event. Like, we've looked in databases. So there are these genomic databases online that are amazing. One is called Nomad. And there are some others like Geno2MP. And these are databases where big consortia or big groups have published not the genomes or the exomes, but they have published the frequencies. So how often you see each genomic variant in different populations, mostly like Europeans and North Americans, but they have about 150,000 individuals. And you can therefore you can calculate how often do I see, you know, these loss of function type variations in each gene? How often do I see every specific mutation? And if we look there, so this mutation here, I think, I might be wrong about this, and I would have to look this up in the papers. Like, it doesn't exist in Nomad, or if it exists, it definitely doesn't exist in homozygous state. So there might be people running around there that have one copy, but that means they still can make ADAMTS19 protein because they still have one functional copy. So the event that you have no functional copy of this, like that you definitely can't make this protein, is extremely rare, and we have not found any other people. There has been one other paper where they have published another family.
But the mutation's not fatal, right?
No, exactly. It's not fatal. Well, at least we don't know. We don't know if it is, right? There was one stillborn here that could have been homozygous as well. But yeah, there's definitely 4 people alive that don't produce this protein, and they're fine. Well, they're fine in terms of the fact that they have nothing except heart valve disease.
I see. So I guess the intuition is that if it's very rare, maybe it means that it's very deleterious, no? That there's very strong
so there's
negative
definitely a relationship.
Yeah.
There's definitely a relationship between frequency of mutation and, so we call that effect size, right? So if you have very rare mutations, they tend to have much higher effect sizes, meaning that their effect is just much stronger. And then very common variants, their effect size on any given disease is much weaker, and they're more involved in polygenic or quantitative traits. So a disease like coronary artery disease, for example, there's hundreds of genes involved in that. And these have all minor effects. So having one given variant might increase your risk for coronary artery disease by 1.3 or something, you know. Yeah. So very, very, or a good example is Alzheimer's, where there's the ApoE variant, which can increase your risk of getting Alzheimer's by three times, for example.
But it could be that there's some other gene that these people also have, right? That fixes this somehow, right?
Like, you mean compensates?
Yeah, somehow. So that if you get this without this compensating gene, you're dead. And maybe these 4 people have some crazy other gene somewhere that like fixes this, right? Is it a mystery that only 4 people that you found have this this thing and are still alive?
No, so it's not a mystery. I think what that points to is likely what Carlos was aiming to earlier, is that actually, you know, a percentage of people that are homozygous knockouts for this might actually not be viable. Might die.
They die before you can sequence their DNA.
They die in utero, yeah.
Okay.
But that's just an hypothesis. We don't know if this is true.
It's not a causal thing, right? It's just like it's just less likely to be born with this sequence, you're saying.
No, you don't. They're not born with it because they would die. They would die in utero. So they would die after the embryo.
I guess my question is, this doesn't cause death, right? Because clearly there are 4 people that didn't die in utero.
Yes, and like you said, like you very clearly and pointed out, is that these 4 people could just have compensatory variants or variation in their genome. That is the reason why they didn't die.
Oh,
Okay.
I see. Yeah.
Yes, they could have something that we call EQTLs that would modify the expression of other genes in utero. Lots of things. There are lots of possibilities of, But what I'm saying is like the fact that we see such few people that are homozygous or such few deleterious variations in this gene is likely due to like negative selection against homozygous individuals with very loss of function variation in this gene.
Makes sense.
Okay, so just to get back to the figure and to finalize figure 1, so this is just basically what they have in their valves. So as I was pointing out earlier, and it would be nice to have a figure for this, but I don't have like a nice heart model or something. But if you imagine your heart, you have 2 atria and 2 ventricles. So the atria pump, and all of this is pumping, right? So the atria pump blood through the valves into your ventricles. So these valves we call the tricuspid and the mitral valve. And then from your ventricles are pressing the blood either via the pulmonary artery to the lungs, and there's the pulmonary valve in there, or from the left ventricle to the rest of your body via the aorta, and there's the aortic valve in there. So those are the 4 valves in the body. And what we've observed in these people, and I'm just going to go to, actually I have to scroll a bit, sorry, to supplementary table 1, because we have a table where we listed what kind of
I remember that table.
Yeah, here we go. So yeah, so you could see that these are the different 4 valves, and some of them had some other cardiac findings as well. But basically, as I said, very early on, these individuals had problems with their heart valves, mostly aortic valve, but also pulmonary. So stenosis is when basically your valve doesn't, it's probably thickened, and because of that, what happens is, think of a garden hose. And when you put your finger on a garden hose, usually the water just flows out, right? But when you put your finger, you increase the pressure and the water comes out much faster. So now this is the same what happens in your heart valve. If the heart valve is thickened, so usually the valves open like this, but then they might just open smaller. So the same amount of blood passes through a smaller opening, increasing the pressure. The reason why
people have high blood pressure, they end up with high blood pressure.
No, actually no, because it's just very localized, right? But what happens is this very strong jet of blood flows against your aorta. So the aorta is like the tube that's on top of the valve, and then basically it increases the stress on that aortic wall. That could lead to aortic rupture, and if you have aortic rupture, that's very, very bad, because if that happens, you usually die inside of like 15 minutes. Like when a young person falls falls over on the soccer pitch or on during sports, and they're dead like 10 minutes later, that's usually like aortic rupture.
Oh,
that's one of the side effects. Then the other one is that this increased pressure causes a negative like feedback loop kind of, and the valve continues to degenerate much faster because of that than it usually would. And so usually these people, when they have that early valve disease, either you have to treat it very like aggressively with medication, or they have to get a valve replacement.
Do they detect it like visually? Like, do they just display some symptoms, or do they have to get some genetic tests to know that it's going on?
No. So you don't need genetic tests for valve disease. What you do is echocardiography.
Yeah.
So if you've ever been to the cardiologist, it's when they put this little probe with some gel on your chest, right? And then that's frequently done. When you go to the checkups for your doctor when you're pregnant, they usually can. Sorry, Perry.
It's like just some ultrasound.
Exactly. Well, so ultrasound is lower frequency than echocardiography, I think. Okay. But it is basically, it's kind of like an ultrasound. Echocardiography is just an ultrasound for the heart, I guess.
Cool. But do they like every day? Do they notice some symptoms, or is it just suddenly your aorta ruptures?
Oh no. So you could have symptoms like fatigue.
Okay.
But it's yeah. So I don't know. It depends on the severity of your valve disease. Obviously, like you could have some pressure in your chest.
Okay.
I'm not a medical doctor, so if anyone watches this,
Lord, I feel weird in my heart sometimes. Can you diagnose me?
No, I can't.
Send you my DNA
I just want to point this out very clearly. I'm not a medical doctor. But this was obviously diagnosed by actual medical doctors, and what you can see just here, just for people that are not familiar with this, so this is an echocardiography where the LV means left ventricle. So we're looking at like the ascending aorta, and this is just the color. What the color means here is the direction in which the blood is flowing. So this is very easy to like kind of mix up, because whenever you see a picture of a heart, usually the color is always like, is the blood oxygenated or deoxygenated? Mm-hmm. So on every drawing of a heart that you see, when the color is red, it means oxygenated, and blue means deoxygenated. But on echocardiography, what it means is it's either flowing towards the probe or away from the probe. So in our case, because
these velocities go like to a meter per second.
Mm-hmm.
Almost
inverse.
Yeah. No, 1 meter per second, right? Almost. Yeah. That's pretty fast.
Is it 70?
No, it's 77. I mean, it's 0.7 meters per second, right?
But it's -1, so isn't it like
it's per second?
Yeah,
per
over
second.
seconds.
Per second, yeah. Okay, right.
So
I made this figure.
That's pretty fast, right? The blood. That's crazy velocity for blood, I feel like.
Yeah, I actually don't know what the standard velocities are.
How many kilometers?
No, I don't know. It just seems like a fast for like.
I mean, haven't you seen Kill Bill, Perry? When they chop off someone's head, it goes like super far.
That's true. That's true.
But it's true. Like, got it right.
You're not wrong.
You're so funny, Perry. I've worked on this, like on hearts for so long. I've never thought about like how fast does the heart, like the blood actually travel through the heart valve.
Oh, I don't know.
These questions are. I took a course on like microscopy.
But it's like 4 kilometers an hour or something, or something. I don't know.
Is it? Do it. Let's do it.
I think it's
Jamie, can you Google that? We have an imaginary Jamie.
You have an imaginary Jamie?
No, I'm just saying. I didn't think. I thought blood was going like kind of slow. I don't know. Whatever. Doesn't matter.
Well, I guess if we would take a heart out of a human right now and we would just cut off the aorta, how far would the blood squirt?
This is a great question.
I'm sorry about that.
We certainly shouldn't do that, but
very cool. So you have a picture of the blood velocity as a function of position.
Yeah. So just to get to the point, what this actually shows is that you have 2 phases in the blood in the heart, which is systole and diastole. So diastole is when the ventricles are filling, and systole is when the ventricles are contracting. So when systole is when blood actually flows through to your lungs and to the rest of your body. And in systole, so when your ventricles are contracting, your pulmonary and your aortic valve are opening, right? Because it comes from the ventricle, so they're opening like this. Then afterwards, when you go to diastole, the valves close. And usually they should completely close. There should be no blood that's going through that valve, because that's the whole mechanical function of a valve, right? It's to limit blood flow into one direction. But in this case, what they had is they had thickened valves. And so you see that there is blood flowing into the left ventricle through the aortic valve during diastole, which is something that shouldn't happen. Yeah, and then this is just a close-up from like on top. Like when we turn the probe, we can kind of look from on top of the valve. So if we take a heart and we cut it, like think of like cutting a heart, cutting the aorta, and then just looking at it from the top, that's what you would see. And what you can just see here is that this is one of the commissures, and it's quite thickened. So white stuff, like white pixels in a echocardiogram, they're white because they're more reflecting the ultrasound. And that's why they're white. Liquid and cavities, they're not reflecting anything, and so that's why they are usually just black or grayish. Mm-hmm. Yeah, and so what this just shows us is that this particular patient that we're looking at here has a very thickened commissure. So telling us that they have a problem with their valves. I mean, these things here, they're clearly telling us they have a problem with their valve. This was diagnosed in a clinic, and this is just to show that, this is what all of those 4 individuals have. Most of them have dysplasia, means thickening. Regurgitation is what I've just talked about, blood flowing backwards. And then BAV is an interesting one, and we talk a bit about it in the paper. So what many people don't know, so usually the aortic valve has 3 leaflets. So it's 3 kind of leaflets that work together to close. So it has 3 commissures, right? Think of like in Back to the Future, you have this, what is it called? The flux compensator.
I see, yes.
I always find that these commissures, think of the flux compensator, right? It's like
okay.
That's what I think about when I think of like.
Jamie should Google that for us.
Yeah, Jamie, he's busy still converting 0.7 meters per second to kilometers per hour. He'll get back to us.
I think 1 meter per second is 3.6 kilometers per hour.
Yeah, I know, but Jamie's a bit slow. You watched the Joe Rogan podcast, Florian?
Well, I don't listen to it, but I know some of the excerpts with like Elon Musk and.
Yeah. He always has a guy who Googles things for him, and his name is Jamie.
Okay.
We pretend to have one too.
You're not quite there with the budget yet?
Not quite.
Exactly.
So
Okay,
basically the clinic detected like this phenotype is weird because these patients are very young, and so they were taken for genetic testing, I guess.
Yes, exactly. So this was is exactly what happened. So these patients were very young. Were seen. I think babies generally, when something's not right, they're seen with cardiologists or in a cardio. And then if something's really not right, then they might go to a specialized clinic. And then they were kind of, I guess, put into like the genetic testing branch of the hospital, and then ended up in the research branch because they probably couldn't find anything that's known.
Mm-hmm. Are the patients authors on the paper?
No.
I think this happens sometimes, though. It wasn't really a joke.
No, I know what you mean. So well, we acknowledge them. We obviously are super like grateful that they participate in the research we do, and it obviously is super helpful when patients collaborate and allow us to utilize their DNA and for research. I think that's great. But authors on a patient, I mean, authorship on I would be fine with it totally. But I think like authorship on a publication is kind of weirdly defined, right? Like,
yeah, for sure.
Kind of have to contribute something to the experiment. So I guess if you are the subject of the experiment, you kind of contributed something.
Yeah, that's always a touchy subject.
But for me, I find like obviously they have the biggest part in this because without them this would have never happened.
Mm-hmm. Yeah, for sure.
I think patients in general, they're just happy when someone tries to figure out what's going on.
Oh yeah, definitely.
Because it is obviously a pretty big thing if your child is born with any kind of defect, like defect, any kind of congenital malformation of the heart, of anything, or any kind of disease, right? And then if no doctor can tell you, well, it's this, you can just say, well, they have this, but we don't really know why.
Mm-hmm.
Specifically, also, for example, in this case, they have 2 children that have this disease early on in life.
Mm-hmm.
You obviously want to know what's the chance of, if I have another child, that this child also has this disease.
Right.
This is, I think, something where, like genetic testing at birth and things like this can really help people kind of manage these situations.
And it's 25% we've established, I guess.
Yes. Yeah.
But now the kids that have it are homozygous, so they're 100% likely to pass on the gene, I guess, or pass on the defect.
Yeah.
The ones that have the phenotype. So they really can't marry each other, basically.
No, but they are 100% likely to pass on one default defective copy. But because it's so unlikely that they meet someone else
that
has a defective copy, usually it shouldn't be a problem. But this is a case where clearly you could say, well, maybe you want to get tested.
Sure. Yeah.
And I think that's helpful for people in the future. Do
you know if oh, sorry, go on.
I kind of would like to know what you think. I think it's actually like we're so getting mixed in terms of ancestries that I guess this is going to be less and less of a problem, maybe, but maybe I'm just in a very multicultural city. And
no, that's for sure. Well, Perry and I both are in interracial relationships.
I'm pretty Mediterranean, though.
Is Greek the same as Italian? I don't know, Perry.
On ancestry.com.ca.com, it's Mediterranean.
What you did your test?
No, but I mean, the categories are there's no Italian category. It's like Mediterranean.
Yeah,
they're all just Mediterranean.
I don't know, but probably we're very probably it's a similar ancestry, right?
Yeah, you should watch out.
We're very similar.
Yeah,
even Turkish. I don't know. Who knows?
Me too, Carlos. I'm also like in a relationship.
You're very interracial, Florian.
Huh?
You're very interracial.
Absolutely. No, but so were the patients happy that you guys found a gene that was linked to their disease? Do you know that?
I don't know that actually, because I don't have contact with the patients because I'm not a medical doctor, obviously.
Right. Must have been cool for them to like learn about that.
Yeah, I think it would be cool if someone would tell me like, oh, I'm doing like research on your genetics and found something cool that's novel. Like,
yeah.
Like for me, the main like one of the joys of doing research and one of the things I think we work for is finding these like observations where you like maybe you're the only one in the world that knows this, right?
Yeah.
Like that's in which like field or which thing can you ever say I'm maybe the only person that has ever seen this?
Yeah.
I don't think
cool.
Very rare, yeah.
So yeah, so this is certainly how I felt with the like next part that I'm going to show you. And then this was like, yeah, I just love these. These are kind of the moments you do science for. And then when you can share that with other people, and that's just finding novel things that we don't know yet. I think that's very exciting.
You did the analysis to find that exome. It was like,
so no,
this one.
So from there, obviously, we have a hypothesis, and our hypothesis is that what the title of the paper says, right? That the loss of ADAMTS19 causes progressive non-syndromic heart valve disease. And we could stop there, and we could say, well, we have these two families, and that's the evidence we have.
Some papers are like that, I think, right?
Some papers are like that, and it's fair enough. If you were a small lab, if I think it's important that there's this trade-off, right, between how good is the evidence to suggest that it's real versus how unlikely is it that it's real. And there's both sides, right? There's papers there where they publish all of these mutations, and there's very little evidence that's and there's very limited evidence that it's true, and then you don't know. And then there's these claims out there that certain genes are related to certain diseases. That's dangerous in a way, and that can kind of flood a bit the science with faulty observations, which then gives science a bad name. But I think it's important, because then if we wouldn't have these observations, we couldn't test them. On the other hand, you could invest some more work to make your observation more solid, right? To add additional evidence to suggest that what you're saying is real.
And that you understand it, right? Like, I think you guys go towards getting to a mechanism of why it's actually causing this like problem, right?
Right. And I don't think we, come up with a complete mechanism at the end, and we still don't know exactly how it works, but I think we've put a lot of effort into trying to understand it better. Yes.
Cool.
Exactly. So I just wanted to go to the supplements, kind of like give an overview of what we've done now, because now it becomes very like deep molecular biology, science, mouse genetics, and I don't want to lose you guys.
No, I'm 100% here.
So I should also start this with saying, like, so we've done experiments in mice, and they're animal experiments, and I know that there's people that are strictly against this, and I can understand why. But for congenital heart disease and cardiovascular biology, it's if you think about it, the heart is a 3-dimensional organ. It's very complex. It's a lot of different cell types that are interacting with each other. The development is extremely complex, and we don't have good functional models that we can culture in a dish right now. So I cannot grow like a 3-dimensional heart that behaves like a heart in the body to test hypotheses like this, right? To play with the genetics and see what happens.
And because this problem is like at a level, like it's beyond the cellular level, right? Like you couldn't just play with heart cells, for example, and figure out what's going on.
Yeah, we could try, but the valve actually has multiple different cell types that are interacting with each other. And it is, exposed to this like physical stress of the blood hitting against it so using any kind of cellular model for that is really simplifying it a lot. And wouldn't really give us the power to validate, whether this gene is actually involved in that. I think so. And I think, that's the whole field is like that. So yes, we are working, like people are developing new cellular models all the time, and they're getting better and better. And maybe in 10, 20 years, we'll actually have like a heart in a dish. That would be super exciting. Yeah. But I'm just trying to point out what the reasons are for why we actually might need, animal models.
Yeah, definitely.
Fair enough.
Yeah. So the type of animal model that we used, it's a kind of like a common model used, which is so-called a LOX-P, or a knockout first model. And this probably looks very like complicated and weird for people that are not used to this.
Could you zoom in a bit? Sorry, Florian, could you just like zoom in a bit so we can see the full figure?
Oh yes, let me do that.
I think you have some plus signs there. There we go. Thank you.
Yeah, here we go. So basically these are three different strains, let's say, of mice. This is what a wild type kind of mouse looks like, and this is just portraying the genomic locus of
wild type means healthy, right? Or
wild type just means like a normal mouse of that strain. So we use mice that are so-called C57 black 6. It's a very commonly used strain in research. They're just black little mice, normal mouse. So they would have, again, this is the genomic locus of ADAMTS19, and we're looking at exon 2, exon 3, and exon 4. This is what the boxes mean. And this line here just means an intron. So again, genetic structure, you have exons, you have introns. So, there's always an intron in between different exons.
Is there alternative splicing going on here?
In this gene, no. In this region, there's no alternative splicing. So there's only these exons.
Okay.
And so this is specifically in this type of mouse, transgenic mouse model, you use an exon that's called a constitutive exon, which means it exists, like it's used in every single isoform.
Mm-hmm.
So it would never be skipped.
Okay.
Because the reason why is what was done, and we haven't done this, but we ordered this mouse. There's a big facility that makes these transgenic mice in Bar Harbor in Maine in the US.
Mm-hmm.
It's called Jackson Laboratory. And they have huge warehouses full of different transgenic mice that they create for their customers and for the research community. It's really amazing, actually. And you can just basically go online and say, I would like to order this mouse that has targeting this gene.
Roughly, how many mice are you usually doing these tests on?
Well, obviously the most that you can, but ethically the least that you have to.
Sure. Yeah.
So we've done quite a bit here, but it's because we've done different experiments, like we've done echocardiography, and you'll see some of the stuff that we've done. We have always this n equals 3 in science, right? I'm sure you're aware of that.
Yeah.
N equals 3 is not really the best for all experiments because it's very limited sample size, right?
What's this n equals 3?
Number of mice you used.
Oh, really?
No. In biology, often you do like three replicates, 3 biological replicates. It's like the minimum that's accepted for statistics and other reasons.
But yeah, don't you have to just have a statistical significance? Like, is 3 enough to give you something statistically significant?
Depends on your type of data, I would say.
Right. Okay. So here you use 3 mice?
Well, we didn't use 3 mice. We used much more.
Okay. Just say the number, Florian.
No, it's somewhere in the table. I mean, it's a lot of mice.
No, but it makes sense. Like I was going to say 3 mice, like 2 families, 3 mice. There's something, it's a small scale thing. But yeah, I guess
let's say there's variation in them. Even if you order like a wild type mice, three of them will be different from each other, even though they're under the same category, right?
But is that
yes.
but is it significance that I guess you have to do the math.
Yeah.
I believe that what you did is correct though. And you used enough mice to prove your point.
But not too many mice.
Yeah, too many mice, yeah.
Carlos wanted a number. I would say if you do something like, for example, breeding strategies and stuff, sometimes you have to prove something that's called like, are these mice born in Mendelian ratios? Mm-hmm. So you have probably heard of Mendel and how alleles are passed to offspring. Mm-hmm. So if we cross, so usually you cross heterozygotes mice. So if I make any kind of transgenic mice and I have, there's 1 copy of the transgene and 1 copy of the actual normal copy of the gene, mm-hmm. these are the mice you would cross, and then the offspring would be born in so-called Mendelian ratios, right? Mm-hmm. You'd have 25% that would have 2 defective copies, 50% that would have 1 defective copy and 1 copy that's okay, and 25% that would have 2 wild-type copies. Mm-hmm. That's Mendelian ratios. To kind of prove that those are correct, you probably need to do at least like 100 breedings.
That makes sense.
Or you need to look at 100 mice, I mean.
That makes sense.
So that's...
You have to couple 100 mice.
No. 100 births are probably enough.
100 births, okay.
Yeah,
not 100 births. Well, 100 pups, 100 mice babies that you need to look at.
Okay.
I would say. I think that's kind of for this type of analysis, for example.
All right.
So it's on the order of hundreds of mice. Fine.
Yeah.
But like each mouse has like 6 to somewhere like between 5 to 10
mm-hmm.
babies.
Mm-hmm. Okay, cool.
Okay, so not to go into too many crazy details here. What the idea of this mouse, transgenic mouse is that, and it's a very standard model that's used again from this company. It has something that's called a LAC-Z cassette, which is a codes for a protein called beta-galactosidase, which can make a color reaction when it has a substrate called XGAL, and basically just turns it blue. So think of this as we put a paintbrush into the cell so that we can paint that cell blue if this gene is active. That's mm-hmm. how you should think about it. Mm-hmm. So we put this paintbrush into the location of this gene, and whenever the cell uses this gene actively, and we give it the substrate, it'll turn blue.
Okay.
Mm-hmm.
It's not reversible, so it'll stay blue, but like it'll turn blue. So we can basically color all the cells that use this gene into like blue. And the reason why we want to do that is that other methods of kind of figuring out where that gene is active sometimes don't work as well. Like let's say antibodies or stuff like that, it doesn't work well. So this is a very standard method to try to figure out when a gene is active during development.
It was very old as well, right?
Oh yeah, this
This
is
method.
like the 70s, 80s, I
Yeah.
think. Before we had a genetic sequence, people were already doing this.
Mm-hmm.
And then all these LOXP sites, they're just there to kind of manipulate the genome. So what we've done then is we've crossed this mouse here with something called CMV Cree. Again, I don't have to explain that. You don't need to understand the details. What it just does is think of, it takes this triangle, and it like removes whatever is in between them. So it removes this antibiotic resistance to neosyn, and this is the important part, it removes this exon 3.
Mm-hmm.
So this is then really creating a knockout. It's creating a deletion, very much like we have in the humans, where they have 8 exons missing. This one has 1 exon missing. But what it leads to, it'll definitely lead to the mouse not being able to produce ADAMTS19 protein.
Okay.
So we're reproducing what we see in humans in the mouse. The fact that we will have mice that cannot make ADAMTS19 protein.
Yep.
And even these mice cannot shouldn't make it and don't make it because they have this very strong splice site. So everything that comes after this blue cassette here will usually never be incorporated and transcribed. So even these mice, they're already knockouts, but this is just an extra step to make sure 100% that they don't.
Okay.
That they can't make.
So you just create mice that can make this protein.
That cannot create this protein, and where we can stain the cells that are actively using this gene blue. Those are the two things. In simple terms, that's why we do this.
Are these mice created with CRISPR?
No, they are not.
CRISPR.
But then I think so. These ones were not. These ones were done by traditional like
they cross them a bunch of times until they have the right.
No.
Genes.
So what they do is they make something. You use a plasmid, which is a round element of DNA.
Yeah.
They use embryonic stem cells from mice, and they kind of insert this plasmid into the genome. Then they inject those embryonic stem cells into pseudo-pregnant mice.
Mm-hmm.
So they then make babies out of those embryonic stem cells, and then they check until they have one pup or a couple of mice where that actually worked, and then they breed those.
What's a pseudo-pregnant mouse?
Oh, that's a good question. They kind of, if I remember this correctly, I've never done this, but if I remember correctly, they kind of like give the mouse certain things to kind of induce pregnancy, but there's no actual like sperm.
Okay. It's an in-between state.
Because they're injecting, or I think actually, no. I think they do impregnate the mouse for real, but then inject these stem cells in place of the actual cells of like the earliest stage of the embryos.
Mm.
Again, I don't want to say anything wrong. This is not my expertise, and I have definitely not done this.
Just, okay. But the takeaway here is that you can create mice that don't have this protein and stain the cell, get some coloring when these genes are active.
Yes. And the coloring, how we do is we take out the heart or the embryo, we fix it with alcohol with a strong like formaldehyde, and then we put the substrate and we do like a thermal reaction at 37 degrees in an oven, and that'll turn the cells red.
So that's in order to get an idea of like when during development the gene gets activated? Like when you want to see a timestamp of when, okay, now they're trying to use this gene, I guess. Okay. Exactly.
Well, and where, right? We really want to know, is it in the valves? Is it expressed in the heart? We don't know this at this point.
Mm-hmm.
Let's do it like a story, right? Like at this point, we don't know if it's expressed in the heart valve. It's just a theory. We've made this crazy claim that there's this gene, and people that don't have it, that can't make protein, have problems with their valves.
Mm.
And just to your CRISPR question, Carlos, nowadays I think these are actually produced with CRISPR.
Mm. Okay.
Because it's such a great tool, they kind of, I think they replaced this very standard. I think they still do the standard approach, but they offer a service that you can make transgenic mice with CRISPR. Because they have a pipeline for that 100%.
Cool.
Because it does make it a lot easier.
I was in a lab that like worked with yeast, and I think it's a lot easier with yeast to make genome changes.
Also, there's no heart though.
There's no heart in yeast. No. Yeah.
But yeast is crazy, right? Because it's like the simplest eukaryote.
Yeah. Yeast people are really into yeast. They love it. They always tell you that beer is made with yeast.
Then I'm a yeast enthusiast too.
There you go.
Okay, so just skipping some of the supplementary figures because I want to first go. Well, I'll start a bit like not in the order of the figures that we've. Let's go. let's go through the order of the figures because it makes more sense if you read the paper like that. So we have these mice. We've breed them now to be homozygous again for knockout for ADAMTS19 so that they basically resemble the humans we have, right? So they cannot make ADAMTS19 protein from birth. We did that, and what we found is that actually, when you look at these mice, 40%, 38% of these mice that are homozygous for ADAMTS19 knockout have heart valve problems. Have
What's the
very
percentage?
similar. Sorry?
I missed that. What did you say? There's a percentage of mice?
Yes, so not all mice. So this is the tricky part. So not all of the mice that don't have this gene, that cannot make this protein, not all of them have aortic valve disease. Only 38% have. However, this is not just like in our model. A lot of transgenic mice models that look at the valves and the aortic valve have this. Like other models, and we mentioned this in the paper somewhere, there are some models that have lower, that have 25%. We call this penetrance of the phenotype. Here we go. So for example, the NKX2.5 mouse, which causes BAV, only 8.2% of mice have BAV. GATA5, which is another gene, only 25% have BAV. Or in a Robo4 paper, it's like 17.9% in males.
But looking at the human, 2 human families, all of the homozygous children displayed this. Okay.
Yes. So in humans, the penetrance would be like 100%.
But we only know about 2 families.
Yeah.
If you measured 100 families.
We only know about 4 individuals. And well, like I said, there's one paper that came out after ours that basically confirms that they have another family with 2 individuals and they also have homozygous, and all these 2 individuals also have valve disease. So it's 6 individuals, let's say.
Okay. 6 for 6.
6 to 6, so 100%. And we don't know why in mice it's only 38%. No one knows. If you find that out, then I'll be very interested.
Well, it wouldn't be as interesting because it's in mice. It doesn't. I guess it's a part of it that didn't translate to humans somehow, right? Like
but one of the challenges is obviously trying to get these models that really, with 100%, we can recapitulate the disease, because it would help us. It was just make our life easier and working with it.
Okay, sure. Yeah.
To have a strong reproducible model.
Yeah.
This way you have to breed, talking about breeding mice again, we have to breed much more mice than we would like because we don't have 100% penetrance.
Right. So then you only look at the mice where they have this, where the penetrance is 100%. Sorry. You only look at the mice that display this problem. That are homozygous.
Well, we look at all the mice, but we obviously note down which ones are affected and which ones are not.
Sure.
Like, for example, here in the plot, you see that we have also some that don't have any problems with they don't have stenosis. So I didn't exclude them from the analysis or anything.
Yeah.
But so this is just again showing. So this is echocardiography now in the mice, and I actually did this. So this is cool to like learn to do.
Those are little mouse hearts.
Mouse doctor.
Yeah. So those are little mouse hearts. The mouse is alive. The mouse is under anesthesia. So we put a little like isoflurane, I think was the anesthetic we used. So the mouse is sleeping. You just shave the chest, and then you can just like you have to shave the chest because the hair would interfere with the echocardiography. But then you, like a human, you put some gel and you can look at their hearts, and it's actually really interesting. So a human heart beats about 60 times per minute, right? You guys guess how fast the mouse heart beats?
Perry?
I'm gonna say quicker.
That's right.
Wait,
So
Jamie, quick.
I'm gonna say 300 times a minute.
Carlos, lower or higher?
A factor of 5. I'm going with.
You're going higher, Perry. I'll take $1.
What? No, I said he said lower. He said lower, higher than 300.
I'll say higher just for fun.
How much higher?
Let me go 1,000.
Oh, okay. So you went too high, Carlos, and Perry was too low.
No way.
So 600 beats per minute in a normal awake mouse.
I wasn't that off, eh?
And in a mouse under anesthesia, it's a bit lower because their heart rate drops. So it's like 450.
I was really close.
So we can't use the same machines we use for humans to do echo. We have
to
have.
So now in the lab, you have like a bucket of mice. You're like giving them anesthesia, shaving their chest, checking their heart.
Yeah.
We're not injecting the anesthesia. It's a little mask you put on their head.
And they're not in a bucket.
Or whatever. You have like a tray of mice. I don't know, a cage of mice. I don't know, whatever. That's nuts. And you're doing it for hundreds of mice.
Yeah, I've done this for tens to hundreds of mice. Yeah.
This took you weeks probably or something. I don't know.
Yeah, years.
Years. Wow. Okay. Very crazy.
And it was really fun to do. Like not fun to do, but like it's a fun like kind of thing. It's like an experiment, right? It's an interesting technique to learn. And I watched,
oh yeah, go on.
I got to go see some, how echoes are done in humans, and the technology is crazy, and it's amazing that we can do this non-invasively and look at the heart, right? I mean,
that feels like real science. I watched this movie on Amazon Prime about Charles Darwin called Creation. It's kind of a boring movie, but it showed like how Darwin was like figuring out evolution, and he had like this shed where he would like collect birds and like fry them so that their flesh would like come off and just have the bones. And he would just do this for like hundreds of birds, just like fry them in some acid or something, and be like, okay, this bone is a little bit similar to that bone.
What?
It's hardcore.
Yeah, it was really intense.
Hopefully he didn't eradicate some of those bird species.
Yeah. That feels, and he like he'd like write it down in like a little journal at night by candlelight, and I'm out here just like writing Python
made a mistake.
code.
Gotta get a new bird.
Yeah.
I just want to point out, no mice were fried in this experiment.
Yeah.
Okay, I don't know if he was frying them, but
so now we have this. The plot thickens. 38% of mice have this problem.
38% of homozygous mice. So homozygous, this is what this notation means, by the way. Knockout. It means both copies are knockouts.
Right.
So you see again, this is like systole and diastole, like I've mentioned before. And in diastole, in a plus mouse, this is not blood that is coming from the valve. This is blood that's coming from the atria, actually. So this is normal, and there's nothing here. But when you look at like, these knockout mice, these here it's the same mouse, has regurgitation. So you see like, this thick jet of blood going. Again, the color is pointing to the direction. So blue means in this direction and red means in this direction. So going this direction. And, in systole, you even see that there's a lot of like blue, which means very fast. So it means direction, but if it's blue inside of red, it just means it passes over the threshold of the machine and it actually goes in the past. So it just means it's faster than what the machine has as a maximum velocity here. Okay. So the blue here doesn't mean it's like flowing backwards inside of the jet. It means it's just very, very fast. And you see that there's like a lot of, the blood is not flowing normal like it should in a wild type mouse. This is basically what this is showing. And this mouse having more of a regurgitation phenotype, and this mouse having more of a stenotic phenotype, where you have very, very high velocities here, like 3,251 millimeter per second. So now you see it's already much less than in a human, which would be expected, right?
Oh, isn't that faster than a human?
No, this is centimeter human. This is millimeters.
This is 3,000. That was 77. This is 3 meters per second.
Centimeters.
Yeah, that's like 770 millimeters. No?
I'm not sure that's true because mouse should be much less than a human, right? Or is it because it's the velocity, the distance actually doesn't matter? Maybe I'm thinking about this wrong.
I'm not sure, but this looks like 3 meters per second.
Seems very fast for mouse. Anyways, This is like, I'm sure this is correct. Like the relative ratio is correct because that's kind of like what the velocities of mice echoes are in general in the literature. And
okay,
but this is very fast. Let's just say this is 3,000. It's very fast. Because in a wild type mouse, you have like 1,000, right? So this is 3 times as fast as you would want it to be here. And this is then when we look at these mice at different time points, you see at 3 months, we barely see, like a difference between wild type and knockouts. 6 months, you see it more. The y-axis here is again a measure that we call the aortic valve peak pressure. So it's just the highest kind of pressure of like that the blood exits the heart by, measured in this millimeters per Hg. And you just clearly see that this is increasing with time. So this is a progressive disease, which is also similar to what we see in humans.
Do you have fewer points for the disease mice? Like, were some of them dying?
Do I have fewer points? I think it's
I don't know.
Like,
I guess they're just more spread out, right?
Looks like the same number of points total.
it just says n somewhere here. How much was
Okay.
it? I think it's one mouse less or so for.
Yeah, n equals 9, n equals 10.
Okay.
Yeah,
Okay.
It's not because a mouse.
Okay. Yeah. I thought maybe that was a thing.
No. Well, it could
Yeah.
definitely be a thing, and there are some mice that died during the course of the experiment, but actually, it wasn't. It was the same it was the same proportion of wild type and knockout mice that died. So usually suggesting that they didn't die because of this
The
disease.
mutation.
But sometimes mice, even like normal wild type mice in a colony, just die.
Mhm.
because, for example, sometimes they scratch their fur very strongly and they cut themselves and then they get infected. Like, and then you obviously have people treating this and taking care of the mice. Like the mice are actually in a facility, right, where they're taken care of. Like they're getting food every day, they're getting their cages cleaned once a week, and
I did in high school for science fair. I tried to do a project about the effects of MSG on what I don't know what, but I like bought mice at the pet shop and fed 3 of them MSG and 3 of them normal food. And so my teacher was like, "Isn't this not enough mice? Like, why would you do this?" And I didn't understand the, like, stats at the time. You know, I was like, "Whatever, let's do this science project," and it was a complete failure.
There you go.
Just like I just wasted 6 mice. Could have been a friend to a child.
Yeah, I don't know if the ethical board would have agreed with your proposal.
That was one of my more ridiculous moments.
Because obviously, when you do these types of experiments, you have to submit a proposal to like the ethics committee of the hospital, of the research center. They have to approve it. It has to be all in compliance with the guidelines for animal experiments and all. Like, this is all highly regulated. It's not just me being like, "Oh, I'm gonna do some experiments with mice. Let's go do it." Like, there's a lot of regulation, a lot of paperwork, and I think it's important and good that it is that way. Like,
yeah, absolutely.
And you have to also, in these proposals, you have to kind of justify, why do you need that many mice? Could it be less?
Interesting.
And so on and so forth. They have to do some power calculations sometimes to say, well, that's what we think the effect size is, and to measure that kind of effect, that's the numbers we need.
Cool.
Okay, so what happens next in this story?
Okay, so final last piece for this figure. I'll try to speed up a bit because I want you guys to be here for 2 hours. If you
Not
look
a
again,
problem.
this is a very similar view than we had for the human. We look on top of the valve, and what you see is that, so this is a normal mouse. You see, like, you know, it's very hard to see, but it's because this is very tiny, and this is actually super difficult to get these images. Technically, I just mean. You see that there's the tesseract, right? These 3. Is it called? What is it called? It's not called tesseract. What is it called? It's called flux capacitor. I don't know why it's the tesseract.
Oh, okay.
I was like,
Where's that from?
Perry.
Tesseract is from Jamie.
From some
No,
sci-fi. From some sci-fi show as well.
This is your specialty.
I watch too many like sci-fi fantasy shows, I think. But again, this is like the triangle. Like, these are the 3 leaflets. Okay, so these are the 3 commissures.
A tesseract is a 4-dimensional cube. It's from Avengers, no, or something.
Yeah, it's from Avengers.
I don't know.
But it's in reality, in geometry, just a 4-dimensional cube.
This is not a 4-dimensional cube.
Yeah.
The point is that specifically this mouse here, it has actually the valve. This one looks exactly like a bicuspid aortic valve would look in humans. So you can see there's only 2 commissures here. So when you see this in humans, probably like a doctor would probably call this like the bicuspid aortic valve. But when we actually looked at this valve, you know, took out the heart and looked at it, it actually did have 3 commissures. So, you know, functionally, it looks and behaves like a bicuspid aortic valve, which, you know, often has these problems like stenosis and regurgitation. But it actually is it has 3 commissures, but they're thickened. They're much thicker. You know, the valve is much thicker. And this is actually when we take out these hearts and we cut them, you know, these valves. So this is what a mouse heart valve actually looks like. So this is a normal mouse. And you see there's a kind of like these thin translucent leaflets. Again, 1, 2, 3, these are the 3 leaflets. And this, when we look at a knockout mice, we actually see that we have this thickening here. You already see that they're much wider, the leaflets, right? The thickening, you don't see it maybe here, but you can definitely see the thickening when we cut them. So here, this is like a cut from through this here in like very thin sections. I think this is 10 microns or 6 microns or something. So this is a normal valve. And now, you know, look at this difference in thickness between the wild type and the knockout. And you see that there's much more these collagen. So the blue here is collagen. This is a stain. It's called a Trichrome Stain. Basically, it's to look at the collagen in tissues. And you can see that there's a lot more collagen in these knockout mice. So this is the reason that we see this functional physiological phenotype when we do echocardiography. This is another stain. It's called a Pentachrome Stain. Again, it's just to visualize different components of the valve. I believe it's also collagen and some other pieces, but it's just another way to look at it and show that there is disorganization So these valve leaflets, what it actually is like they have like a layered structure of something that we call extracellular matrix, which are just proteins like keratin. For example, like keratin and stuff like these extracellular matrix proteins give structure to your tissues.
Mhm.
And in these valves, it's the same thing that's happening. But what happens in these knockout mice is that this is disorganized. Like they're loosely combined. You know, they're not packed together and very organized like they should be.
Mhm.
We don't know why. We just observe that this is the case.
It's interesting that like the disease phenotype in both measurements like shows a crazy amount of variance, right? Versus the wild type. Like
What
it's
do you mean
almost
variance?
like I'm looking at the bar plot at the bottom there.
Yeah.
You have like I mean, it's almost like a uniform distribution, right? Across this scale.
Why is it uniform? Like there's much fewer points.
But they're all like
Oh, yeah.
almost equally spread, right? Like there's as many like close to the wild type as there are at the extremes.
So the
Well,
reason
there's
for
2 at
this
the
is,
extreme, and then 5 or 6 at the bottom, right?
well, so I can tell you the reason why this is like, so these are obviously like some of, this is the thickness of this here, right? This is what
Yeah.
we're quantifying.
Mhm.
So you have 6 measurements per mouse, right? Because you have well, you don't see it here, but like basically you have 1, 2, 3, 4, and then 5, 6, and you don't, in this cut, you don't see the other 2, but basically you have 6 per mouse.
Okay.
And in some knockout mice, not all of these are thickened. Not all of the hinges, this is what we call hinge.
Mhm.
Not all of these hinge like arms, they're thickened in each mouse, just some of them.
Mhm.
But yeah, it's crazy, right? It's weird. Is that not the whole valve swells up like crazy?
Mhm.
And it's a question we're asking ourselves, whether there's like preferential thickening of certain leaflets due to the position in the heart, It's definitely a question we are asking, but we couldn't see a pattern.
Mhm.
Because probably our N was too small.
More mice.
Would need more mice for that, yes. Yeah, okay, so we're at the point now, we've made a mouse model, the mouse model observes the same disease the humans are observing, but we haven't yet figured out whether that's due to the gene that we're messing with, right? We're kind of assuming it is. But now we're going to look at like, okay, where's this gene actually expressed? Well, actually, I'm going to skip over this. This is just a more detailed look at like what the phenotype actually looks like, what the tissue looks like. So this is electron microscopy. So it's just like better microscopy, more resolution, right? And again, you see just that in a wild type, you have this layered structure where, and I'm going to zoom in a bit, this layered structure where it's very organized layers of like collagens and extracellular matrix and cells. And then when you look at the disease, you see, it's night and day difference in terms of the structure. So this is just showing again that the extracellular matrix is completely disorganized. You have a lot more debris. The cells actually look different. So, something's not right with these valves. It's simple. Okay, and now we're going to look at like, where is this gene or protein actually active, right? So now we're going to look at, in this figure, we looked at 3 different time points. We looked at 15.5, which is so E15.5. So mouse pregnancy takes usually about 21 days. So this is, and the heart is usually formed completely at like 14.5. So 1 day before this, the heart is fully formed, and afterwards just grows.
But this is while the fetus mouse.
This is a fetal mouse. Yeah, E means embryonic. So this is from an embryonic mouse.
It's in the belly.
In the belly. Okay.
Yeah. So we have to take out unfortunately, we have to sacrifice a pregnant mother and take out the embryos.
Ah, okay. You don't just ultrasound. Oh yeah, I guess you can't go through the mother to check out the heart.
Yeah, I'm not sure that it's probably possible to take out the embryos without having to sacrifice the mom, but that would be a super involved
yeah.
It's like
team of 12 surgeons for one mouse.
Stat.
Yeah, and so basically, so we look at 3 different time points, like Carlos was saying, to kind of figure out when this gene is actually active, right? What you can see, so this is what a mouse heart looks like. What you can see, again, these are the atria here, these little pockets, and then these are the ventricles. And this very thick, like dark dot here, that's actually where the valves are. That's actually the aortic valve, or the pulmonary valve, if we look into this. This is a pulmonary. So, like this, we obviously cannot see super great. We can see that it seems to be active, in the atria and in the valve. But we can then do sections. So again, we can take it and put this heart into like wax and then cut very thin slices of it, like you would cut bread slices, but just much thinner. And when we do that, again, we have the 4 different valves here: tricuspid, mitral, pulmonary, aortic. And I want to focus on the aortic and the pulmonary. What you see is that, like I said, this blue color is always super active in the valves, right? The pink color here are cells that are not positive, so that are not having this gene actively working or like actively transcribed. And the blue cells are genes that are actually using ADAMTS19. Right. And what we see is that we see inside of the valve leaflets, exactly where we would expect it, we see this very, very strong activity of ADAMTS19, confirming that this gene seems to be very important in those types of cells and in the valve tissue, and actually in all 4 valves. Mm-hmm. And I want to show you a picture from actually one of my favorite pictures from the, and we've done this, we've also looked at embryos in earlier time points, and you have very weak activity. For example, here at 10.5, so 5 days earlier, you have activity in the cartilage, but actually almost no activity in the heart. But and then at 12.5, you do have activity in the heart, but very weak in the valves. But I want to focus on this one here, where here it's big at 21 days after birth, so postnatally 21, you actually can see the valves by eye, and that's maybe it's a better way to visualize this for you guys. Mm-hmm. So again, if I take the heart, I cut now here on top of the valves, I remove the atria, then I cut off part of the ventricle, I can put this heart like in the dish and look from the top onto it. And now you can really see all 4 valves here: the aortic, the pulmonary, the mitral, and the tricuspid. And I think you can beautifully see here that these leaflets, this tissue is blue. Right. Therefore, it's positive for ADAMTS19. So, this goes full circle with, we had a gene in humans. We thought it was involved in valve disease. We made a transgenic mouse. The gene is expressed in the tissue very specifically where we think it has a role, and the mice recapitulate the phenotype.
So in figure 3, you already have some evidence, right? Statistical evidence that the lack of this protein, or the inability to create this protein, is correlated at least with higher chance of having this thicker. Yes. I don't know what are we calling them again? Thicker valves or whatever, or thicker valve walls, or I don't know what you call them again.
Yeah, exactly. Definitely. So we've never observed any wild type mice with these defects, but we've observed 38% of the knockout mice that we tested, which were like, I think, a total of, you've asked for numbers earlier. Let's go to the numbers. I have to go down, sorry. We have those numbers. So we've done like a lot of mice and tested a lot. Let me see. Here we go. So these are the Mendelian ratios. And then here, so out of 77, 29 had some defect of the valve.
Right. And then, so this is statistical evidence already that there's some correlation between the production of this protein or the lack of the gene that can produce this protein and this disease.
Exactly.
And then in figure 5, you're showing now that actually this protein this gene is actually directly influencing the valves somehow, right? There's activity in the valves.
It actually is, directly is expressed there during development of those valve tissues, which, suggests that it probably is important. Like if it's active there and then you remove it and the valves are not working correctly, it seems to have an important function, right? Mm-hmm. Yeah, so that's the
punchline.
Well, the punchline is that, and then I could go into like the very cool like last part we've done.
Yes, I see a T-SNE. Oh no, it's a UMAP?
It's already a UMAP, yeah.
Yeah, very nice.
This was like right when like T-SNE became, let's call it uncool, and UMAP became cool to do. I mean, both of them are valid dimensional reduction techniques, I think. And both of them have advantages and disadvantages.
Yeah, for sure. Oh, I mean, let's look at this one. This looks nice.
Okay, so it's the last thing I'm going to talk about. But so we kind of came full circle to, okay, we really believe this gene, ADAMTS19, is the one responsible for this disease, but we don't know what it does at all, right? And we don't know how it's regulated, and what is actually causing the disease, right? We just know this gene is not there, mice get sick, and humans get sick. But why? Why is that the case? So what we've done is, this was in 2000, I think that when I did this was 2017, it was right around the time like single cell technologies in biology became really interesting, fancy, and became really like more easily doable because of advances in like molecular biology and the technologies like with it. And we've actually done this with a technology called DropSeq. So we've built our own machine back in the days that would enable us to take like a heart, dissociate it into single cells using like enzymes and some mechanical dissociation. So you take a heart, a mouse heart, you put it in this like mix of enzymes and into a machine that can physically dissociate it. And then you end up with like a liquid cocktail that contains all of the different cells of the heart.
Mm-hmm.
So it will contain cardiomyocytes from your ventricles and atria. It would contain fibroblasts, endothelial cells from your coronary arteries. And it would also contain the cells that would be found in the valves. So we've done this for wild type and knockout mice. And the first thing we wanted to look at again was to prove, which we already knew, but just another type of assay that would prove this, that ADAMTS19 is actually expressed in this particular cell type in the valves that's called the interstitial cells. They're kind of mesenchymal cells. Those are the cells producing the extracellular matrix. So those are the cells critical to kind of making the construct. I
don't get it. You have this cocktail, and what are you doing with this cocktail?
Oh, this cocktail of single cells. Sorry, yeah, I cut completely in there. So you have this cocktail of single cells. The way that most people do single cell genomics these days is you use microfluidics. So you put this cocktail of cells through a machine that has microfluidic channels in them. And what you do is you capture the cell in a little oil droplet alongside a bead. And in our case, these are, I haven't worked with DropSeq in a while, so I don't remember what the beads are made of. But it's basically tiny like beads that are coated in like in short primers or nucleotides.
Okay.
And these can capture polyadenylated mRNA. And there's like a lot of fancy words, but what it just means is in your cells, the RNA that gets actually translated into proteins, they have a little like tail, which is called a poly-A tail. It's just lots of A's, lots of adenine. And there's a reason for that stability during export from the nucleus to the cytoplasm. It's a signal for the cell to know this should actually go from the nucleus to the cytoplasm. But we use that as a trick to fish out all of this RNA and put it on the beads. So these beads, they have long stretches of T. And by complementary binding, these long stretches of T will bind to the long stretches of A from the cell. So what happens first is this: when the cell enters a droplet, it gets lysed. So it explodes. There's lysis buffer in the droplets. So the cell explodes. All of the RNA is now in this droplet, and then because of diffusion and just thermodynamics, these RNA with the A's will end up with the bead at some point and will hybridize due to complementary binding. And then we can use those beads. The bead has a barcode, so we load these cells into this microfluidic machine at a very low rate. So what happens is we only have one cell with one bead and one droplet. In most instances. Again, the speed, we use this with some molecular biology technology to, at the end of the day, be able to get the RNA, sequence the RNA from each individual cell. We sequence them all together, but because we have a barcode, we can de-identify them afterwards.
I think the normal thing is that like you take a whole bunch of cells and you get DNA sequences for that whole thing. So like when you're doing normal sequencing, is you don't know where like that gene is, like what that gene looks like in different parts of the body, right? Or different cells. Maybe that gets to Perry's question earlier, right? If like this mutation was present in the whole body or in specific cells.
Yeah, so traditionally, Perry, we would have taken a whole heart and we would have just mushed the heart together and then sequenced all the RNA from the heart. Like that's what they would have done 10 years ago. Or maybe we would have cut out the valve, smushed the valve together, and then just looked at that one. But that would have only given us one observation. That would have been all the different cell types mixed together.
Right.
With single cell, it allows us to really look at each cell type individually because we're sequencing every individual cell.
I see.
So instead of having one observation for tissue, we have like 10,000.
Okay.
And they differ in their biology because they're different cell types. Like you see here, there's lots of different cell types in the heart, right?
Right.
And we are specific.
How do you know which tissue each cell came from when at the moment of sequencing?
Well, this is one heart, so I know all of these cells come from a heart.
Oh, sorry. Which cell type you're sequencing?
Good question. So we don't know. All of these cells are just dots that have specific profiles, right? But we use we try to identify genes that are very strongly expressed in one group of cells, but not the other.
Okay.
This is basically what this heat map is showing, right? Like if you look here, this group of genes here, ADAMTS19, collagen 9A3, and TGF-beta inhibiting factor, they're all very highly expressed in this cluster 1, which is VIX, but they're not expressed very strongly in any other cluster, indicating that they're very specifically expressed. You try to find like topics, I guess, that are very specific for one
It's
group
like
of dots.
K-mean, what's it called, Carlos? This machine learning.
K-nearest neighbors.
K-nearest
K means
neighbors.
clustering. No, sorry, K means clustering.
That's exactly what we use to define these clusters here.
To get the colors, I guess. But to get the position of the points, you did this UMAP thing, which is like
Yes, based on principal components. So you do a principal component first, and then you use the first 30 principal components as the input to your.
Very
You could
cool.
also
but
use
a point here is like
a cell.
a cell, but what genes it expresses, I guess, right?
Yeah, but not all of them. Just you traditionally use only the most variable across all cells.
It's a principal component to find or whatever.
But like
Yeah.
the raw data is like a vector of like which gene is this gene expressed or not, sort of. Am I getting that? Or
It's
is it
a
like
big
raw
matrix.
sequences? Okay.
No, it's a bit matrix of counts of integers that is genes in rows and cells in columns, or cells in rows and genes in columns. But it's a matrix of, let's say, 25,000 times however many cells you have.
Right.
Yeah.
In our case, it's a 25 times 50,000 cells, I think, approximately, is what is in this plot. Yeah. So again, it's a lot of background that you kind of need to know to understand this, right?
Yeah.
So it's hard to know how much I need to explain about different things. The basis of what this plot is trying to show is like these are hearts from different mice that we have pushed through this method, and we can identify different cell types. How do we identify them? We identify them by these genes. So we would call them marker genes because they mark specific cell types, and they're very distinctly expressed in that cell type, right? And the point that we were trying to make is to say that ADAMTS19 is a marker for this specific cell type, which is valvular interstitial cells, which are the cells producing the matrix of the valves. And I think we can clearly make this point here. Then we use the technique. The next thing we wanted to know is what is ADAMTS19 actually regulated by. So gene expression, works via transcription factors, and genes are regulated in a coordinated manner. And we use some techniques. We use a program called Scenic here. What it does is it just looks kind of at co-expression of genes. So for each cell, it says, is this gene expressed in the same level than this other gene or this transcription factor? So it has a list of transcription factors, and then it will figure out which genes are probably co-regulated by that transcription factor. And this kind of led us to this transcription factor called LEF1, which I'll start with D. If we look at all of the different transcriptional programs in these VICs, then this is the most specific, meaning this is the one that is the most specifically only expressed and only active in VICs and not in most of the other cells. So this was exciting. And why do we look at this? ADAMTS19 is actually one of those 111 genes that are predicted to be regulated by this. And it's super specific, right? So now we've identified a transcriptional program that's super specific for this one cell type, and our candidate gene for this valve disease phenotype is regulated by this gene, saying that probably this whole program is pretty important for valve development. And we actually validated that, this is responsive to this, and there's a plot in the supplementary material that kind of proves this. Okay. So now we have this gene. We know we have an idea what it's regulated by. The last thing we wanted to look at was like what is actually going on in these valves. So we've looked at knockout valves, knockout cells from knockout mice and normal mice, and we compared them in this specific cell type and in the cell type that's covering. I'm going to zoom here. And in the cell type that's covering the valves. So like I said, the cells that we looked at are those cells here that are blue but not red, right? These are the VICs. But the other cell type in the valve are endothelial cells, which are lining, so the monolayer of cells that's lining the interstitial cells. So these have usually like a barrier function, right? They're protecting the other cells from being exposed to the outer world. And we actually found that those are the cells showing a strong signal in knockouts. So those seem to be the cells that react first and actually have even though the gene is not expressed in them, right? The gene is expressed in those cells here that are blue. They're not blue because of the fact that we stained them. This is a different, technique. This is DAPI staining. The blue here are just nuclei. But I'm just saying, this is the area of the valves where these cells that are positive for ADAMTS19 would be found. So that was interesting that like the cells where the knockout happens, they also have differentially expressed genes. So they also have a signal, they have a response, but the stronger response that's probably actually driving the phenotype actually happens in these cells that are not expressing the gene. So what we think it's going on is that lack of this gene actually causes this layer here that I was telling you, what we've seen, causes the structure to be disorganized. And therefore, the valve is kind of unstable in terms of the physical structure it should have. And because these endothelial cells are the first cells that are experiencing that unstableness, the instability, they are having a response. And this is what we test here. So we looked at a shear responsive, transcription factor, so a gene that responds to when the cells have more stress. And this is highly upregulated in the endothelial cells of knockouts, but not in wild types. So it seems that this is really the kind of driver of disease and
mechanism.
Yeah, well, the part of the mechanism. It's probably not all of it, but pretty important piece.
It's crazy to me that you guys reconfirmed the same thing like 3 times. Like it's really no. I mean, crazy in a good way. Like it's very like I feel like you guys tried everything you could to try to prove your hypothesis.
Yeah, and I think that's what happens when you have a review process. Certainly we try to make very, very sure that we, and I think that's always good in science, right? Like you try to make sure that you can reproduce. You try to make sure that you're very, very certain and sure of what you're.
I'm just saying I would have like been convinced, I think, even after figure 3. No, I mean convinced that something was like there was some relation. But then you clearly show in figure 5, I guess, that like, look, this is like, there's even more than just like a correlational effect, right?
But I think you could keep going even further, right? And see like, well, what's like the structural, at the level of the individual molecule, like what's going on, right?
Yeah, I'm sure you could push us to the infinity,
yeah.
right? Like, and then just figure out exactly what the protein does.
Yeah.
Yeah, but I think it's really cool.
Yeah, I hope it was interesting for you. And I do realize that it's very different from the other journal clubs you've done.
I believe very strongly in diversity, and I think this is very good.
I mean, you also went through it very carefully and very, I think it was good the way you presented it.
I loved it. I really did.
As did I. We would invite you on whenever else you wanted.
No, I'm happy you guys like it. I mean, it's obviously been like a lot of my work that I've done. It's been a couple of years that I've spent on this. And yeah, I think it's a really interesting story. I'm still really fascinated with this gene, and like you said, like Carlos was hinting at, you can go much further and try to find out. But it's hard because working with these models, like I said, then not all of the mice get sick, so it's a lot of work and a lot of time, and so you have to have the right tools to answer these questions.
But you got a nature genetics out of it, so that's, I think, time well spent.
How was the review process on that?
Very long. If you look at when it was submitted and when it was accepted, it's like a year now.
Also, it must be very long to like, for all the collaborators to like have their say or like read the reviews, or I don't know what you guys do there.
No, I think that is not the longest part. I mean, everyone has been super collaborative and been.
I mean, I'm just
working
saying,
really great together on it. But you always give them 2 weeks at least, right? So yeah, you send it 2 weeks.
Then it's like everyone has to make comments, right? Or not everyone, but probably a portion make comments that you have to implement, and then it's like it probably extends the process.
Yeah, of course, all of the authors see it and comment on it and help with the writing process.
Yeah.
But there's I would imagine like a lot of these experiments, the reviewers ask for them, no? Or some of them.
Yeah. There's a lot of experiments where, I mean, obviously can't say which ones, but there's obviously a lot of, the first version of the manuscript that we submitted obviously looked very different to this one in terms of, there's a lot of experiments that were added during that's why it took so long. Because when you go and do these mice and wet lab experiments, you have to order the reagents, you have to plan the experiment. In the terms of mice experiments, you have to breed the mice. So yeah, certainly like papers like this where you have functional experiments take much longer than bioinformatic papers where all you have to do is change a bit of code and rerun some analysis.
Which I like already find super annoying. I can't imagine like, all right, we've got to kill 100 more mice now.
Let's order some more mice.
Oh my God, I have to go back to my code now. Are you kidding me? I'm not doing that.
Shave more chests.
Do this.
Yeah, it is. I mean, it's challenging, and it's one of the reasons why I personally didn't want to do more experiments, because it's
No, but I think it pays off in the end, right? Like you did like real science, which is cool.
I think bioinformatics is real science too, though.
I mean, sometimes I don't know. I think there's a lot of variations on it, but you're looking at the physical world out there, which is cool. I mean, now I think you can do that. You're right. You can do the same thing sort of on a more virtual level, but it must be very rewarding.
It was like, certainly this was a great project, like a very fun project to work on, and yeah, I'm super happy with the paper again. I'm super happy with everyone who worked on that, and I think everyone did a great job. Again, it's teamwork. It's teamwork. So I'm talking here, but I think I talk for like hopefully everyone who worked for this.
It's not just have a podcast with them.
Yeah, send them the video.
Yeah, sure. And then they'll all tell me that I said things wrong or something.
Well, they can put a comment, and we'll have a very.
We'll have them on next time. The whole consortium.
counter arguments.
Debate.
So what are you working on now?
So now we're working on, mostly I'm working more on quantitative phenotypes. So like I said, this is like Mendelian, what we would call Mendelian genetics, right? Like you have 2 copies, you're sick. Now I work more on traits that are, I'm actually trying to look at the quantitative nature, or like the quantitative genetics of valve disease, let's say. Because, so yeah, you can have these cases where it's a rare disease, very strong mutation, you get a phenotype, but there are also, genetic variants in the genome that just increase your risk of getting valve disease when you're getting older. So getting it a bit earlier or getting it more severe.
Mm-hmm.
And so I'm trying to look at that with tools, there are these tools, like these things that we call genome-wide association studies. I'm sure you heard of that, Carlos.
Yeah.
Yeah. So trying some of those things to figure out whether we can find new variants that are involved in that. And then just learning a bit more about like statistical genetics and by and large, I think that's been really fun. Really enjoying my postdoc with Guillaume.
Mm-hmm. Nice. So this was all before your postdoc started.
Yeah. So some of the review process was while I was already in my postdoc,
mm-hmm.
which is challenging as well, because then you're kind of in a new job and you have to, new responsibilities, but you still have to kind of figure out these things that are related to your old job.
Mm-hmm.
But yeah, so this is work. Obviously, Gregor, as the last author, he has been my PhD supervisor. Mark is a good friend of mine in Germany, in Kiel.
Mm-hmm.
And yeah, it was a long but very interesting project.
Excellent. Well, thanks so much, Florian. Any questions from you, Perry?
No, I think I asked them during the presentation, and everything went to. Yeah, thanks for giving us explaining this. So it's like giving us the details.
Thanks for asking the questions.
Yeah, of course.
Oh yeah, thanks for your time, Florian. Had a lot of fun.
Yeah, for sure.
Thank you, guys.
And if ever you want to do this again, let us know.
Yeah, definitely. I'll definitely be down. Take care, guys. Have a good Christmas.
Thanks a lot, Florian. See you later.
Merry Christmas.
Yes, Merry Christmas. Special Christmas episode. Bye, guys.